आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सोल्यूशन पाओ। डाउनलोड नाउ।
Hello everyone, so here's the question. Question says name the source organism that possesses the Taq polymerase. So they are asking about the Taq polymerase. What is so special about the function of this enzyme? So what happens in the polymerase chain reaction, that is PCR technique? PCR stands for polymerase chain reaction. Now in this technique, basically what happens, we take a small section of a DNA, a very small section of DNA, and this section of the DNA, it is being amplified approximately 1 billion times so that we can study that DNA, and that for study of that DNA we use this technique, polymerase chain reaction. Now in this technique, the first step of the technique is denaturation. And in the denaturation, what happens, we take a strand of the DNA and at a very high temperature, that is it is being provided at the heat, and you know that DNA is a polynucleotide change. So what will happen? It will separate. So two strands of the DNA, they will separate in this technique. They will separate in this step. All right. So this is 3 dash 5 dash, over here 5 dash 3 dash. Double stranded DNA has now been separated. Second step, after the denaturation, what will happen? The second step is known as annealing. It is called as annealing, and in the annealing process, we add primers. We add primers to the separated DNA strands, and you know that function of the primer is to initiate the replication. So now after the addition of the primers, third step will be replication. The DNA, the primer, it will start replicating, and after this step we will add this Taq polymerase enzyme. Okay, this Taq polymerase enzyme, and this is basically a deoxynucleotide. Along with this, we will add deoxynucleotides. So along with the replication, there will be extension. So third step will become extension of both the strands of DNA. All right. Now this cycle, this is being performed approximately 30 cycles. Okay, this is being repeated like 30 times. So 30 cycles will be there. 30 cycles will be there, and hence it will result into the amplification. It will result into the amplification of this DNA strand. Amplification of this DNA strand. So actually we wanted that small strand of the DNA, to study that small strand of the DNA, we wanted to amplify that DNA, and for the amplification we used 3 steps. First step was denaturation, second step was annealing, and third step was the extension. When the primers were attached and they started multiplication in the different, that is 2 separated DNA strands, we added an enzyme that is known as the Taq polymerase. This enzyme, Taq polymerase, it is being obtained from the bacteria, and the name of the bacteria is Thermus aquaticus. Thermus aquaticus. Now, why we use specially this enzyme only, polymerase enzyme? Because Thermus aquaticus bacteria, it lives in an environment where the temperature is more than 100 degrees Celsius. And you know that any enzyme, if it will be put at the higher degrees or higher temperatures, then what will happen? Denaturation of the enzyme will occur. So to prevent that to happen, we use Thermus aquaticus Taq polymerase. Why? Because this bacteria possesses an enzyme, that is the Taq polymerase, which can substand even at the higher temperature. This enzyme will not go under denaturation even at a higher temperature. So over here, what is so special? It will not undergo denaturation. This enzyme will not undergo denaturation, and hence we use this Taq polymerase. Thank you.
क्लास 6 से 12 से लेके नीट, IIT, JEE मेंस और एडवांस के लेवल तक 10 मिलियन से ज़्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।